So Helena just presented on surface temperature and methods and theory behind that. And now I'm going to follow up and present background on measuring hormones non-invasively in saliva, and kind of use the same framework. There'll be some theory and then really kind of best practice guidelines, or at least recommendations for how to measure the hormones in saliva. So everybody can see the slides, I hope. Everything's working well. So just some general background to remind everyone that hormones are chemical messengers that diffuse from glands, endocrine glands, that are located throughout the body. So here you can see the approximate location of the endocrine glands in pigs. And they can have actions at distant sites from where they're released. And I think most of us know this definition of hormones, but we don't always think about what that means. And practically, that's really interesting because it basically means that hormones, when they're released peripherally, are measurable, detectable potentially in any bodily fluid, and that they can have actions throughout the body in a lot of different areas, depending on receptors that are recognizing those hormones and where the hormones can bind. There's three main classes of hormones. The peptides are sometimes called protein hormones, and these are mainly synthesized in the hypothalamus and some in the pituitary. They include the oxytocin, which we'll talk about a lot, and other hormones like corticotropin-releasing hormone and adrenocorticotropic hormone. And these are really important in the hormonal cascade that leads to the release of the steroid hormone cortisol, the glucocorticoids. And these hormones can be after they're released, they can move into cells, and they can actually be stored in cells and then released later when needed. So that's one special property of that class of hormones. And the second main class of hormones are the steroid hormones. And these hormones are synthesized from cholesterol prior to their release, and they can't be stored. So really all the available hormone has to be synthesized and then released. And the main steroids that people know about a lot are the glucocorticoids, including mainly cortisol in most mammals, corticosterone, which is the main glucocorticoid in rodents and also exists in other mammals, and which is synthesized in the adrenals. And then the sex steroid hormones, estrogen and testosterone, which are predominantly produced from the sex gonads, the ovaries and the testes, but are also produced in smaller concentrations from the adrenals, which is why everyone has circulating estrogen and testosterone in their bodies. And the third class of hormones that we're not going to talk about too much, but it's good to know, are the catecholamines. And these are modifications of the amino acid tyrosine. And so ones that people are probably familiar with are epinephrine and norepinephrine, or adrenaline, sometimes and noradrenaline it's also called, that are also produced by the adrenals. So that's just background on the main classes of hormones. And hormones from different classes have different properties. The differences in stability, differences in the ease of measurement. So it's something that I'll keep on reminding you about when we're talking about the main hormones that we're measuring, which ones are steroids and which ones are peptides, and that has implications for how you try to measure these hormones. So the biological effects of hormones are determined first by the concentration of hormones in circulation. And so you can see here, this is showing you plasma oxytocin in cows before and on their day of parturition, when the calf is born. And you can see this huge increase in the circulating concentrations of oxytocin right around, really peaking at the birth process, and then returning not to baseline, but reducing. And so this is really showing you the huge change in the concentrations of hormones that are happening to facilitate important processes. But it's not just the concentration of hormones that changes. For biological effects, the strength of the biological effects is also determined by the presence and the number of receptors in target organs that these hormones can bind to. Because hormones, as I mentioned, need to bind to receptors to have their effects. So I think what people who maybe think more about the peripheral physiology, Arador is so aware of, and why it's really good also to collaborate with neuroscientists to do this research, is that it's really fascinating to know that the receptors for hormones also can change, both in terms of their density and their distribution in the body and the brain. So here you see in cows again during pregnancy, the steady increase in the number of oxytocin receptors in the endometrium in these cows as pregnancy increases. And same thing, you have this kind of peak in the oxytocin receptors around the time of birth. And this is showing you that for a really important process like the birth process and a successful birth, it's not just increasing concentrations of hormones, but it's actually changes in the number and the distribution of the receptors for those hormones in target organs that are important. And then hormone effects are also mediated by negative or sometimes positive feedback loops. And here's a nice illustration from a recent review on oxytocin and maternal behavior that's showing an example of a positive feedback loop. They're less common, I would say, than negative feedback loops, but they also occur. So here you have the suckling behavior of the piglets stimulating the teats, which are sending then messages to the brain, which are stimulating the posterior pituitary to release more oxytocin, which is synthesized in the hypothalamus, released by the posterior pituitary. And from the posterior pituitary, the oxytocin concentrations increase in the circulation. And this also facilitates more maternal behavior, like nursing behavior, which then has a positive feedback and releases more oxytocin. So in this case, maintaining elevated levels of oxytocin is part of facilitating the lactational phase for pigs and other mammals. And just a really important point that hormones aren't having their effects in isolation. They're interacting with other hormones, also other biomolecules. But I think that's something we don't always have the capacity or think about how to deal with, but we should really not measure hormones in isolation. We should try to measure them also and see how they're connected with other hormones, because the other hormones really can influence, especially the negative and positive feedback loops. So when we're measuring hormones peripherally, I said that the hormones are available in a range of bodily fluids. Also, some hormones can be measured even in the hair follicle. So we have to decide which matrix we're going to measure the hormones in. And when we're making this decision, we have to think about a few considerations. First of all, what's the acceptable level of invasiveness? And not just acceptable for us ethically, right? But also acceptable in terms of being able to accurately assess the responses of the animals and not be influenced, not have the animal's physiology too heavily influenced by our actual measurement of their responses. And so we typically think of measuring hormones in blood as the most invasive. Of course, this typically requires restraint of the animals, which is really stressful for them, and also puncturing the skin, which can, requires a high level of training, and there can, of course, be problems with this. And arguably, the other matrices are minimally invasive, but why I show this kind of range of low to medium invasiveness is I think this really depends for saliva, for urine, for feces. It really depends on your methods for collecting these samples. So you can do sampling in a way that's going to truly be minimally invasive in the sense that the animals aren't going to be restrained and they're hopefully in their home environment. But you can also, if you're restraining the animals or if you're separating them socially for long periods of time to get these samples, I would consider this to also be moderately invasive and potentially affect the hormones. So you have to think about the methods, even when you're looking at matrices besides blood. You also want to think about the desired time frame for detecting changes. In blood and saliva, you can see changes peripherally after specific stimuli in relatively short time frames. So within 5 minutes in blood, within 15 minutes in saliva, you can see changes. And also typically the peak and return to baseline can happen in under an hour, although of course this depends on the intensity of the response, right? But in general, you can measure very quickly changes in the physiology. In contrast with urine or feces or hair, you're measuring concentrations of hormones that have accumulated over more time, from a few hours to even a few weeks. So you want to think about the time frame. So these might be better if you're looking at more long-term measures like mood, where blood and saliva are good for really short-term emotional assessments. And you want to also think about if you want to use a very established method or if you're going to try to develop your own method for measuring the glucocorticoids, and I would say many steroid hormones. There's really established methods in many matrices. For measuring oxytocin, which we're going to focus on today too, which is, if you remember, a peptide hormone or a protein hormone. These, in general, the peptides and the protein hormones are harder to measure peripherally. We do have validated methods, but no widely accepted gold standard. And that's confusing for people first coming into this field, and also confusing for us who've been working in this field for a while. You'll see this, I'm going to really show you explicitly, even with pigs, measuring oxytocin in saliva, it's done really differently across different labs. And so that's really something to be aware of. There isn't currently a agreed upon best method. And that's a problem, of course. That's something that we need to change in the future. Good. So we're going to focus on saliva today, which is the matrix I measure hormones in, mostly in pigs. So the pros to using saliva is that you can collect samples from animals in their home environment, in their social groups, without specialized training. Of course, there's different techniques that you'll use for different animals, but it's really can be very can be done by any personnel without a lot of specialized training, and the animals can stay in their home environment, which is great. And I would argue, this is my personal view, it's even better than plasma sampling for detecting short-term changes and long-term patterns in some hormones. So let's say for sure for the steroid hormones, where we have really established methods in saliva, I can't think of a reason why plasma would work better, because if you want short-term changes, the restraint and the taking the sample itself it's really hard to make sure you're not affecting the hormone measurement, because changes in the plasma happen in such a short time frame, and the restraint of the animal and taking the sample can take a few minutes. So I would be nervous about affecting the measurement with the restraint. And for long-term patterns, unless you put a catheter in your animal, it's really hard to get repeated samples over long periods of time. It's just highly invasive. So with saliva, you don't have that problem. So at least for some hormones, I would say saliva is even better than plasma. For others, we still have to work on validations. And there are reliable, commercially available kits. So there are kits that are validated for saliva. Not usually in pigs, usually in humans. So it means you still need to validate it for your species, but at least there's validations for salivary measurements of hormones in humans, which is a good start. So the drawbacks for saliva, and I think it's stuff we really need to think about and talk about more and do more to address. We actually don't totally understand how some hormones reach saliva. We know we can measure these hormones in saliva, like oxytocin, but it's not totally clear how they get there. And this is confusing. We also know very little about how salivary hormones are influenced by other biomolecules in saliva, like antimicrobial components, other proteins in saliva. This is an area that we just need more research in. And so this leads many to question, what do we really measure when we measure a hormone in saliva? And this is from 2005, basically a publication saying oxytocin isn't a valid biomarker when you're measuring it in saliva by immunoassay. The opinion on this has generally shifted since then, and the methods have improved, but it's still an issue. It's still something we need to really think about, especially with the peptide hormones. So let's focus in on the glucocorticoids. They're the steroids, evidence of steroid hormones or examples of steroid hormones. They are activated when animals face challenges. So they're activated by the sympathetic nervous system during arousal or in coordination with the sympathetic nervous system in situations of arousal. And so they're released from the adrenals after this hormone cascade. And then they're actually mobilizing resources to deal with challenges. So in the short term, cortisol and glucocorticoid release is a very adaptive response that's important for dealing with challenges. But we also need to return to homeostasis. We need to restore reduced levels of cortisol, and that's why we have a lot of these negative feedbacks. So cortisol is providing negative feedback higher up in this hormonal cascade, and ACTH provides negative feedback to then signal, okay, enough, we're dealing with the challenge, hopefully we don't have to release more cortisol. And if that system is dysregulated, and if we don't have these checks on cortisol release, then we can get this chronic elevated, or actually hypo or hyperregulated. So high levels or chronic low levels of cortisol, which is very maladaptive for animals. And that's when we have to worry about inability to return to homeostasis. And the other thing that I think is really critical to note is that when we talk about cortisol release to deal with challenges, we're typically thinking about negative challenges like a predator threat, but cortisol is also released in almost in similar concentrations in response to some highly arousing positive challenges. For example, sexual behavior in rodents triggers cortisol release, at least in the males. So this is what cortisol is doing in the body, and what's happening, how is it getting to saliva, so that we can measure in saliva. So after cortisol is released from the adrenals, a lot of the cortisol in the blood is actually bound to proteins called corticosteroid binding globulin, CBGs, or other proteins. And so the cortisol is moving through the system, but the bound cortisol is harder to measure. And it's not passing to the saliva, but the unbound cortisol, so the remaining unbound cortisol, can pass through by diffusion, cell diffusion into the saliva, and it can then be measured in saliva. And what we hope, what this implies, first of all, is that only a small fraction of the total cortisol that is secreted in response to a stressor can actually be detected in saliva. But what we hope is that this unbound or free cortisol is representing well the biologically active fraction, the cortisol that can bind to receptors. So the bound cortisol is not biologically active at that time. And usually in some situations, the free cortisol and the total cortisol or the bound cortisol are correlated. Not always the case, but there's differences in opinion on this, but in general, there's this belief that, or hope at least, that the free cortisol in saliva may actually be a more accurate representation of the cortisol that is mobilized to work in the body to respond to challenges. This is even more complicated because there is some CBG, some of these binding proteins in saliva, but in really, really small amounts. So maybe 10% of total salivary cortisol is expected to be bound. So the majority of salivary cortisol is free, whereas the majority of plasma cortisol is bound. And this is something that is important to keep in mind and may influence also how you want to, your decisions about how you're measuring these hormones. So we have some really nice biological validations showing that what we're measuring when we measure cortisol in saliva is corresponding with what we'd expect and what we can measure with cortisol in plasma. So for example, salivary cortisol increases very quickly in response to known triggers of cortisol release. And this includes brief social isolations of young pigs, and also what you see here is social defeat. So this black bar here is showing a situation where an individual loses an aggressive encounter, a pig loses an aggressive encounter, and what you see is very quickly increases in concentrations of cortisol in saliva, shown in the C figure, in plasma, shown in B, and also increases in ACTH, the precursor for cortisol. So you have coordinated increases in cortisol in all of those matrices, and then a gradual return to baseline over about an hour. So that's a really nice indicator that yes, what we're measuring in cortisol is also reflecting what's happening in plasma. And we also can look at cortisol concentration changes in saliva in response to exogenous cortisol agonists that we're administering. And one of the most common ways to do this is with something called the ACTH challenge. So here we're injecting this precursor, the initial steps in the hormonal cascade to release cortisol. We're injecting that into animals, in this case, also into cattle. And you see at the time of injection, increases in salivary cortisol, which is in the black bars here, and corresponding increases in plasma cortisol concentrations in the gray. So we see that if we apply an external agonist, we get the expected changes. So in general, cortisol and other steroid hormones are really well validated to be measured in saliva. So let's move on to oxytocin, which is, as we mentioned, a peptide hormone. It's actually also, it works as a neurotransmitter in the brain. So oxytocin is synthesized in the hypothalamus. It can be released peripherally to the brain, where it binds to a lot of social brain centers to facilitate social recognition and memory. It interacts with the dopaminergic reward system in the nucleus accumbens, and it binds to the amygdala, where it's reducing fear and anxiety, which then can promote social interactions. And not only is oxytocin working as a neurotransmitter, but of course, then when it's released from the posterior pituitary, this is part of the endocrine system, and now it's acting as a hormone in circulation, where it can interact with the sex gonads and the breasts and coordinate things like sexual arousal and orgasm. You saw the link to the birth process and lactation, and also provides feedback to the central nervous system. What's complicated is that central and peripheral release of oxytocin, they can be coordinated, they can be correlated, but they're not always correlated. And this is something that's makes it complicated to know when we're measuring oxytocin peripherally. Is it actually reflecting what's happening in the brain, or is it something very different? That's important to keep in mind. But we do see that, at least in some situations, the increases peripherally in oxytocin reflect what seems to be what we'd expect oxytocin to be doing also in the brain, which is responding to salient, mostly social stimuli. Not always, but mostly social. And this is why there's a lot of interest in this idea that maybe oxytocin peripherally could be an indicator of positive emotions and used for positive welfare, because we're really looking for these indicators of positive emotions. There's some promising evidence there, although I think there's a lot of negative results too. So that's important to say. This isn't reflecting all of the evidence, and we don't see robust changes in peripheral oxytocin in all of the positive contexts that we look at. A good example is play. We just don't get peripheral oxytocin changes to play in pigs, even though they're really playful. And it's clearly a positive behavior for them, but we don't see this. You also don't see that in plasma. So some of the situations we expect to evoke oxytocin, we don't see it. Other situations that are more promising is some positive human-animal interactions lead to increased salivary oxytocin. This is an example with pigs who are receiving more stroking from a human caretaker who they know, who they like, have increased salivary oxytocin. And we also have some evidence for this in horses with training, with a positive training experience. And also, interestingly, this is just a study that also came out from PIGWEB that was just recently published, and showing that pigs are having higher salivary oxytocin concentrations when they have been trained to expect a positive reward. In this case, the receipt of straw at a certain time every day, where they hear an indication of a sound and they know they're going to get the straw. And then this is the salivary oxytocin concentrations averaged before and after they have their straw reward. And here's on the right side, the salivary oxytocin concentrations where they expect this positive outcome. They think they're going to get the straw, but then they don't get the straw in the end. So it's this violation of expectations. And here you see lower salivary oxytocin. So that's an exciting finding that I think we need to follow up on, which is interesting because it's not explicitly social. And it's showing you that oxytocin isn't only working in social contexts. It's also triggered by food and sex, and well, sex is social, but food and some other positive contexts that aren't explicitly social. Okay. So we have this potential to measure oxytocin peripherally. Maybe sometimes this can be reflecting what's happening in the brain, which would be really exciting, but we know that's not always the case, and that makes it complicated to determine this relationship, as I said. And we also have to figure out what we're measuring when we're measuring oxytocin in saliva, and that's also more complicated because oxytocin can't diffuse through the cell membranes like cortisol. It's lipid insoluble. So for a long time, we didn't understand how oxytocin could be getting in saliva, but people kept on measuring it there, and it was definitely getting there. And so that led to this idea that oxytocin is passing into saliva from blood capillaries through a process called ultrafiltration, where you just basically see that example here. It's basically slipping in through these junctions in between cells. And that's the established idea about how oxytocin is getting from blood into saliva, but there's still a lot of questions about this. And so an important implication when we're thinking about oxytocin in saliva is that we don't have such clear correlations with oxytocin in plasma and definitely not in brain. And we wouldn't necessarily expect such good correlations as we get with steroids. It doesn't mean it's a problem, but it means it's harder to do some of the validations we'd like to do and to figure out the relationship between oxytocin and saliva and in other matrices. And we also don't, just like with other hormones, but even more so with the peptide hormones, we really don't know how they're being affected by salivary parameters. And this includes things like differences in flow rate of saliva, which are probably influencing ultrafiltration. So it's much more likely with peptide hormones that the amount of saliva in the system, the flow rate, is influencing how much is passing into saliva. But I don't know of any studies that are really looking at this carefully with oxytocin. It's something we need to look at more. There's also less agreement in how oxytocin should be processed, both before measurement and the best ways to purify the sample if that's needed before measurement, and the best ways to measure the sample. And there's fewer good commercially validated ELISA kits for salivary oxytocin, but there are some really good ones. So that's a good first step. So in terms of biological validations, we know that salivary oxytocin increases in response to known triggers of oxytocin release. And here you see an example from Lopez Arjona and colleagues, showing that when male boars are ejaculating at this time 0, salivary oxytocin is significantly higher than before ejaculation, which we'd expect because we know that oxytocin is involved in sexual arousal in males and females. And here's a study from our lab showing that we were actually able to collect saliva samples from sows during their birth process, which takes hours usually in sows. So there's more than enough time to get in there and get some samples. And we got samples at 5-minute intervals during the birth, just to see if there was variation, maybe spikes happening somehow during the birth. We see a little bit of variation, but mainly we see that salivary oxytocin is increased during the birth compared to the day after the first day of lactation, and also compared to before birth. So that's also a validation of increased salivary oxytocin related to processes that we know are mediated by the oxytocinergic system. We don't have too many biological validations where we're manipulating agonists or antagonists of oxytocin and seeing effects on concentrations in saliva or in blood, actually. So we need more of those. But one validation that we did was we used high-performance liquid chromatography, or HPLC, which is able to detect specific compounds based on their physical properties. And what we basically showed was that what we'd labeled as oxytocin in pig saliva that we'd extracted and measured looks like it has the same physical properties, so that's in the blue, as the pure oxytocin standard that we're using that comes with the kit. So this fact that they're basically showing up in the same fraction of the HPLC suggests that they're the same thing or have the same composition. So that's a good validation, but we need more validations, biological validations of oxytocin and other peptide hormones in saliva. Okay, so that's the first half. That was kind of the theory. So now I'm going to move into the how, actually. So how do we design studies and collect the samples and analyze the samples to get the most out of the potential to link physiology to behavior and welfare. So, so these are my recommendations. Of course, this will depend somewhat on your own personal goals for your study, but in general, I recommend using a within subject design for your study in terms of your measurement of hormones. That's really important because individuals vary both in their baseline concentrations of hormones and also in how much they react to challenges. And it's better to use an individual's own baseline as the reference point to see how extreme their reaction is. This is true whether you're looking at short-term emotional responses. In that sense, then you'd want minimum of 2 samples per subject, right? You need a baseline sample before and then a sample after the stimulus of interest. And of course, you need that for every different stimulus or different condition that you're interested in testing. And if you can get more samples, it's even better, right? If there's, in some ways, going in and collecting saliva can disturb the animals in terms of their interactions. So you have to find the balance. But if you have more samples, you can characterize not just the initial increase, but maybe how long the hormone, how long is required for the hormone to return more to baseline. Same idea for long-term processes. So if you're interested more in looking at basal concentrations and trying to characterize kind of homeostasis, then you want to measure the hormone concentrations within an animal at different time periods, maybe different developmental periods, maybe when they're in different environments, and see how that influences it. And here, the recommendation, not just for me, but also in the literature, is at least 6 samples per subject. And that's really also a minimum, get at least 6 samples. Some people are collecting 1, 2 samples and trying to say something about the baseline conditions of these animals with hormones, and it's really limited, I think, what you can say with that because of this variability. So those are some ideas for sampling design. Then when to collect your samples. So if you're looking for acute responses, the short-term responses to events, you want a pre-stimulus sample shortly before the event. I would recommend 30 to 5 minutes before your stimulus. It can also be longer, but if it's closer to when you apply the stimulus, that's great. And then your post-stimulus sample, we mentioned that changes in saliva happen quite quickly. You want to get that sample about 15 to 30 minutes at latest after the stimulus. And of course, if your stimulus lasts longer, then maybe you can, sample a little bit later. But in general, the peak response to the stimulus should happen within 15 to 30 minutes after. If you're interested more in basal concentrations, this is an example from a study we did. So in gray, you can see the different days that I'm collecting actually on those there's 2 saliva samples from the pigs. And so I'm collecting 2 samples because I want to actually match the timing with when I'm collecting pre and post samples from the other stimuli that I'm interested in. And I'm collecting them across different days of the pigs' lives. You can see that, so it's not just on one day. And I'm taking into account things like the circadian rhythm of the animals, how long they've been awake, how recently they fed. All of these things can influence the hormone concentration. So we're trying to have samples collected across a wide range of time days, but at the same time between 9 and 9:30 in the morning to control for these other factors. You can also collect the samples at different times of day, but you really want to control for that in your models and in your analysis. Okay, so in terms of the actual materials to use for collection and also storage, I recommend using swabs that are actually made for saliva collection. That we have a lot of these for infants and children, a lot of companies that are making swabs specifically for saliva collection that can be then used for animals. And these swabs should have a synthetic material. So cotton can interfere with hormone measurements. I really don't recommend using cotton at all. Use a synthetic material. And the other reason to use a synthetic material and these swabs, especially made for saliva sampling, is that they're more absorbent. So you're going to get more volume of saliva per time period collecting compared to something like cotton. Your collection and storage tubes also really matter. Try to use polypropylene tubes and avoid plastic or glass for short-term and especially for long-term storage. And the reason is because there's evidence that hormones can sometimes bind to plastic or glass, and then they don't always can't always come out from that. So you can lose some of the concentration of the hormone if you're using those other materials. I recommend whenever possible to have a styrofoam box with ice chips in it and keep your collection tubes on ice while you're collecting. It just stabilizes temperatures. It's not so important for cortisol and other steroid hormones because they're relatively stable hormones, but it's really important for hormones like oxytocin and other peptide hormones that are less stable and can be influenced by temperature changes. I recommend at least 3 habituation sessions, separate habituation sessions with the animals. This is the way to, Helena also talked about the same idea. You get the animals used to you if they're not used to being with humans and used to the process of giving the sample. And you can always kind of make this more appealing to them by giving them a little, dipping the swabs in applesauce or something they'll like if they're hesitant, if they're scared. Only do that once or something during the habituation. Don't continue with that, of course. We don't want the animals to associate the swabs with anything especially positive, and you probably won't need that, but it's an additional tool to use just during the habituation if needed. After 3 habituation sessions, most pigs will have no problem to give you a sample voluntarily. And I'll show you what that looks like in a second. Safe sampling, just be careful, make a handle. Don't get your hand near the pig's mouth. Everyone who works with pigs knows why that's important. So we tape the swab really securely to some wood dowels. You can also use a surgical clamp, but then you have to clean that in between different samples from different animals. Typically for our experiments with the within subject design, We, of course, want specific samples from specific individuals, and so we need to make sure that each sample is specific to an animal that's usually identified somehow with a back number. Try to avoid food or water to the pigs or your subjects for at least 15 minutes before collection. It should ideally be 30 minutes to an hour, but we found realistically in a farm setting where these pigs are not typically food or water restricted, that 15 minutes is what we could handle. But just be aware that if food or water right before collection, it can influence the samples. It has to be recorded if it happens, and it can have an effect. Attempt to keep the swab in contact with the mouth for 1 to 2 minutes per subject. Of course, this varies by age and animal, but you can estimate about 100 microliters of saliva per minute the swab is in contact with the mouth. You don't have to have the swab in contact with the mouth continuously for 2 minutes, but in total 2 minutes will give you about 200 microliters, 0.2 mils. That's a good amount for trying to measure oxytocin. You'd want 300 microliters really for oxytocin. So it's a little, I think in 2 minutes, you can get about 300 microliters usually. You should check that. And for cortisol, you need much less. So it also depends on which hormone you want to measure, the volume you'll need. And always attempt to place the swab on the side of the mouth under the tongue. And the reason is because that's where you see the salivary glands here that are secreting saliva. So if you're near those areas, it's more likely that you'll get a nice wet sample. Okay, I'm just going to show you a video here of a collection of a saliva sample. Now, these pigs were very mellow when I came to do this. It's not always so easy, but this is an example. When the animals are habituated and they're familiar enough with you, and you move slowly and try to not disturb them, it's usually not too hard to get the pigs to chew. So this was also, I mean, that's not usually the case that being filmed. We had a film team in there recording for another reason, but I thought, oh, I should show them this because now people will think collecting saliva samples is really cool. So if you collect saliva samples, maybe it'll get filmed. But so the samples, it's in the back of the mouth. The pig's chewing on the sample. I'm putting it on the other side now. You want to do that for about 2 minutes. And yeah, that's it. So, of course, sometimes it's harder. Sometimes the animals are avoiding you, and then you need to have a little patience, but usually you can get a sample pretty quickly from each animal. After you get the sample, you want to inspect it, discard it if there's any signs of blood. You can really see these red spots if there's a cut in the mouth or something. This will interfere with your measurement entirely. Feces or food on the swab is not a good idea if it's very obvious that it's contaminated. Ideally, you want to centrifuge the samples as soon as possible and within an hour in a centrifuger with a cooling function. Aliquot them into smaller tubes and keep replicates. Don't put all your volume of saliva in one tube because you want to be able to potentially measure multiple hormones or at least repeat measurements on a hormone, and you don't want to have repeated freeze thaws of the hormones just for stability. So just aliquot into a few tubes. And store the samples at -20 or preferably -80 degrees until analysis. And that includes if you're shipping them somewhere. You have to ship your samples on dry ice. You don't, I mean, I guess for steroid hormones, you can maybe ship them without that, but I wouldn't recommend it. And for oxytocin, you really need to keep the samples frozen if you move them. And try to analyze your samples within 9 months of storage. I mean, even in -80 over years, the concentrations can degrade. Okay, so now to the more, I would say, controversial parts of the oxytocin measurement. So there's really a lot of disagreement and ongoing questioning about whether an extra purification is needed before you actually measure the hormones in saliva. And the idea is that this extra purification could maybe remove a lot of interfering biomolecules, other proteins, other things that might interfere with your measurement. And In general, for steroid hormones, this isn't needed. I don't know of any procedures that do a purification step for salivary hormones and steroid, and for salivary hormones when they're steroids. For peptide hormones, it really depends on the species. I mean, there's actually different properties of the saliva in different species that can influence whether it's needed, but it also depends on the lab. So in our lab and in a lab of our collaborators, we run solid phase extractions, which is a pre-measurement purification step for salivary oxytocin measurements in pigs, but other labs measuring salivary oxytocin in pigs argue that this step isn't needed. And this is just something interesting that we have to resolve when we get to this, closer to this gold standard. A solid phase extraction, in brief, is basically you load your sample, the saliva, onto a column, and then you're basically doing a series of washes, often in a mildly acidic solution that's supposed to wash out the potential contaminants, other proteins and biomolecules, while maintaining your oxytocin in the cartridge itself. So the oxytocin is stuck there, it's not coming out. And then you use, at the end, a more acidic solution that's eluting out your analyte of interest, so your oxytocin in this case. And so the arguments for it is that it's removing potential sources of interference, and it's also important if you want to concentrate your samples. Sometimes your oxytocin is just in low concentrations in saliva, and your kits won't be sensitive enough to detect it unless you can concentrate it. And the solid phase extraction is a way to concentrate samples, so it's also used for that. And of course, the drawbacks are that it can induce new sources of error. So I understand why people don't want to do this if they don't have to. The main thing for you to know is if you're going to measure oxytocin or other peptide hormones, you really should check yourself if a purification helps. And the way to do this, it's actually very simple. Start with your unextracted sample, start with your saliva. You just collected the saliva, it's stored -80, and now you want to measure oxytocin. So take those saliva replicates, make a pool of a bunch of different samples, so you have a consistent one solution, and measure that in replicates. So 8 times, 10 times on the same plate. If you're using an ELISA, we'll get to that in a second, but assuming you're using an enzyme immunoassay, just measure the sample in replicates. And you should get an acceptable level of similarity in the concentrations of this pool. And the second thing you should run is a parallelism, which is a linear dilution of your sample, and compare it to a linear dilution of pure oxytocin, which you can get with a lot of, you can buy it on your own or get it with kits. And these are showing here for horse saliva, at least, that we have a really nice parallel slopes of the serodilution of oxytocin in pooled saliva samples and a serodilution of the pure oxytocin in blue. For us, for pigs, we can also get a nice parallelism, but only if we spike the sample, because the concentrations were too low to run a parallelism without spiking. And for us, that's the reason that we extract our samples. It's also for the concentrating of the sample. Check yourself, especially when you have a new species, if you're first measuring oxytocin in saliva, you need to check and see if these validations work with unextracted samples. And otherwise, try an extraction. And there's a really good literature on the different methods for solid phase extraction for oxytocin. There's a lot of similarity in the methods. I just recommend checking for your species, for your samples, what works best for the wash solution. So you want a wash solution that is slightly acidic, but not eluting oxytocin. So we use a 10% ACN wash solution. And you want an elution solution that's eluting all of the oxytocin, but not getting stronger than that. So we use a 60% elution solution, which is eluting all of the oxytocin in our saliva. So you can check this yourself, and this is from a publication that's describing how we check that. So methods for salivary hormone measurement. The original, or I would say the earliest measurements were done with radioimmunoassay or liquid chromatography mass spec, or LCMS. They're still the most precise methods, but they have drawbacks. They're quite expensive, and they, in terms of radioimmunoassay, you have to use radioactive materials. So I think the most common method right now is enzyme immunoassays, or ELISAs, and here, this is basically relying on this specific antibody-antigen interaction. So you have antibodies in the kits that are binding to antigens in your hormone. And when they bind, the reaction produces a detectable signal. It's most commonly a color change. And so here you can see what this looks like after the analysis on this Aya plate here, where the darker colors are indicating that there's more analyte, more antigens that were bound in the sample. There's a few different ways of doing of ELISAs, but that's the basic idea behind it. And then you have to read this in a microplate reader. So you need a specialized machine to analyze it, but that's when you have this machine, it's great, and you get nice readouts of the concentrations of your hormone in each sample. And a similar process for measuring cortisol as well. You just don't need to worry about this pre-measurement purification step. There's also a really interesting technology. I honestly know very little about it. If any of you are working with this, I want to talk to you after. It's called Alpha Lisa technology. And so this group, Lopez Arjona and colleagues, are using this technology to measure oxytocin. And it's based on laser excitation. It's really interesting. They're also getting manifold differences in oxytocin concentrations in saliva than we are. And that can happen with different methods, but it also it makes it confusing to figure out what are actually biologically relevant concentrations of oxytocin in animals. So it's an interesting technology. I think we should keep an eye on it and think about it as well. So just the wrap up basically, run validations of your measurement. And these include some we've already talked about, the precisions, so repeat measurements of your concentration of oxytocin in a pooled sample, the parallelism or linear dilutions of the oxytocin in saliva compared with the standard curve. And this is shown here, the validations for the kit we use, which is from Cayman Chemicals for salivary oxytocin. And so you can see their validation of oxytocin in saliva compared to the standard. And also recovery when you're measuring the amount of oxytocin in samples that you spiked with a known amount of pure oxytocin. These validations, you can't just use the assay that has validated it, although it's great to have an assay that's done these validations for oxytocin, but then you also, of course, have to validate them in your lab, or make sure the lab you're working with is doing their own validations. That's really important. And then there's also validations you do on every plate that just include basically checking that replicates of the same pool across plates give you consistent measurements. And we need more biological validations. They're critical for establishing that salivary oxytocin is a reliable marker. So we should do more tests of salivary oxytocin changes in response to known triggers of release. We need more working with agonists and antagonists of oxytocin, like we have with cortisol with the ACTH challenge. These biological validations are going to lead to greater acceptance of non-invasive alternatives. And that's what's sad for me to see. Sometimes I think people are really excited about trying to adapt non-invasive measures, and I'm really happy to see that. But if people aren't setting up their study in a really well thought out way, and if they're not doing thorough validations, the results aren't going to be trusted by other physiologists. And so if we care about these methods becoming more widely accepted and actually figuring out what are the limitations and the potentials, we need to also be doing the physiological assessments and the validations. So it's just my plea for more about that. 5 minutes left. That's going to be fine. Just a little bit about best practice approaches for analyzing these wonderful samples that you've collected and that you've measured now. And so sometimes I see that people have this within subject design, but then they just average. So they've taken the pre and the post and they're averaging it somehow. Don't average your data. Keep this within subject repetition. Use all your samples. Keep the outliers in. They're also really interesting. And if you have enough data, you can run linear mixed models. I recommend the LME4 package in R, and it can deal with all of that. It can deal with the within subject repetition. It can deal with outliers. And this is just a very basic idea about how you could run your statistical analysis with your cortisol concentrations as the response in pink. You're basically an interaction between the context that you're interested in, baseline versus positive or positive versus negative, whatever the context was, and the sample type. Was it the pre-sample or the post-sample? And when you interact these things, you're going to measure whether the change in cortisol differs based on the context. And that's how you can integrate in this fact that you have this within subject pre and post ability to actually look at individual reactivity. Put in control predictors in orange. They could involve rank, sex, definitely the time of sample collection, which we know influences things, and put in random effects in gray here. Random effect for subject, because you're repeating it. You have repeat samples from the subject. A random effect maybe for each group that the subject was in, the day of sample, etc. And then here is this showing this result. This is for the PIGWEB project, where we have a joint manuscript we're working on actually across several institutes. And this is cortisol changes from pre to post sample in a negative context, and the same pre to post sample in a positive context. Here you see what I would say is the negative context was more arousing for the animals, but you have an increase in cortisol. You don't see that the positive context was arousing. At least you don't see the change in cortisol. And this is an example of applying this interaction to look at changes in reactivity. And I highly recommend thinking about your data that way. So in summary, non-invasive measurement of hormones in saliva is a really good tool, both for short-term assessment of physiological reactivity and for long-term assessment, where you can get repeated samples over time in minimally invasive ways to look at homeostasis. For steroid hormones, I'd argue saliva is better than plasma. I don't see any reason to use plasma anymore, really, for steroid hormones. For peptide hormones, we need more validation and standardization. And those are really the future prospects. This is just really briefly to show you, just for pigs, this is also from a publication recently I have, so you can look at it there, but this is showing you the variation in the collection materials, whether extraction was performed or not, what measurement method was used. And those are the main things that we've talked about today that are varying a lot between labs just for measuring salivary oxytocin in pigs. It's really the next step is to get all these people together and figure out what's going on with these different measurements. Maybe they're all basically showing the same patterns and it's just different ways of doing it. Well, we should really check that. And finally, as Helena said, for the peripheral temperature, it's just as true for hormones. Emotions are multi-componential processes. Your data analysis should be multi-componential as well. Link your hormone measurements with other behavioral measures, with other physiological measures, and you can use a lot of approaches to do this. This is showing a principal component analysis for this PIGWEB project, where we have in here the change in oxytocin, the change in cortisol, and other behaviors that we're linking to negative or positive events in the animals, so we can see with the PCA how these different measures are relating to each other. And I think that's really important for the future of improving on and expanding on this work. So that's it for me. I also have the citations, and yes, I'm happy to take questions now. I know it was pretty tight. We just have 10 minutes left. I don't know if it actually turns off in in 10 minutes, but in any case, I'm happy to take questions. Also, I'm sure Helena would help to answer questions as well. So please shout them out or write in the group. I know it's a lot to take in. I see that people are writing about the recordings. There's going to be a recording and there's going to be a PDF of the slides that will be sent to everyone. Let's see, I'm getting. No. Any questions also for Helena? Also for me? No, everybody's gone. Are there anything that was really surprising or new for people?
A lot of thank you in the messages. They find that the webinar was really interesting and important. So
Okay, well, we're gonna hang out here for a few more minutes. If anyone has questions, I'm really happy to answer or go over anything again. Are there any aspects that you would want to know about? Oh, there's a question. Yay. I was also going to ask if there's aspects that people are interested in that weren't covered here. So that would be, I think, also great to know what else people would want to know that wasn't covered. So what about saliva sampling versus fecal sampling? Yeah, it depends on, sorry, there's another question too, but I see this. It really depends on your question. So I've used fecal sampling for cortisol in field settings where there was no way to get a saliva sample, right? You can't get close enough to your animals. So if you're working with wild animals, feces or urine, definitely go for that. Also, if you're interested in accumulated concentrations of hormones, like a really interesting way that fecal cortisol is used with primates in field settings is, let's say there's been a huge upheaval in the social group. So you have a new male come into the group and the whole hierarchy is shifted. What researchers have done is use the fecal cortisol samples from before. So they were already collecting kind of baseline samples. And then they got fecal cortisol during and after this upheaval. And you could really see how stressful the upheaval in the social system was and which animals were the most affected. So that's a really nice use of fecal samples in a time when saliva wouldn't work. If you have the choice between both, I would say saliva is much better for short-term emotional reactions. And fecal sampling would be better for more long-term, the kind of hormone levels over days or at least hours. Can't see if anyone's raising their hand actually, so you can just shout out.
Laura, yeah, has a question. Yeah.
Hello to everyone. First of all, thank you very much for this webinar. So interesting, really. Thank you. Well, I have a question about the courses you're offering for this next January and February, because I am really interested to attend to one of them. Especially, I believe the question is if, for example, if I do the course regarding to the scientific research, that will help me to also to use the camera in the veterinary clinics. because I have to choose one. So,
Yeah,
so
yeah.
that's why I am asking that. I
Mhm.
can't do both. I
Yeah.
mean, I hope to do one at least.
Yeah, yeah.
That's
There is.
the idea.
Yeah. Thanks. Really nice to hear that you are interested, and that you are interested in both. There is quite a lot of overlapping content as well. So both of, regardless of which course you come into, you will learn a lot that you will be able to use either in scientific or a veterinary setting. But just to make sure that you can find the course that is the best fitting for you, could you please send me an email, and so we could discuss in more detail. So that to find, so if I hear in more detail that what are the situations where you are planning to use it, then I will be able to determine which course will be more useful for you.
Okay. See, yes, of course. I
yeah.
will. I don't know. May
And
I?
you can one way of contacting. Did you see our website, the Arador website, where the link goes? There's also, yeah, there's also a contact form. So if you scroll down on any on any page on the website, including the course website, if you scroll down, you will find a contact us link.
Okay. Well,
Yeah.
thank you very much.
So yeah. So if you
will.
if you fill in that contact form and send it, so then I will get your email address, and then we can discuss through email in more detail that which course will be better suitable for you.
Okay. Well, thank you very much, Helena.
Thank you.
And I have more questions now in the chat. One about recovery. So I'm going back to that slide. So I would say that for ELISAs, the main analytical validations that every lab should do are the parallelism, precision, and recovery. And they're asking, can I explain recovery more? And basically, the idea with recovery is you take saliva where you've already measured the concentration of the hormone in the saliva. So you've already measured the oxytocin in that saliva. You have extra samples from that same measurement. And then what you're going to do is spike different replicates of those samples with a low, a medium, and a high pure oxytocin. And you can get guidelines for it. You basically want to spike within the range of the assay measurement. So from the highest to medium to the lowest values that you could expect to get within the assay. And then you're going to spike the sample, and now you're going to measure it again by ELISA. And what you'll see then, you can calculate you'll get the actual oxytocin in those spike samples, and you can calculate the expected oxytocin, right? Because you know how much your initial sample, what concentration of oxytocin was in your initial samples that you measured, and you know exactly how much pure oxytocin you spike the samples with. So you compare the actual concentration that comes out when you run the sample with your expected concentration, and there's an acceptable range. You basically want to have 90 to 110% recovery of oxytocin in these different samples. And so that's the measurement of recovery. And there's really good step-by-step, I would say, descriptions of how that can be done when you look at enzyme immunoassay validations. So there's some good papers on that. Some of them I think are in my reference list, so hopefully you can find that. And we did, PIGWEB actually had a summer school where we went through these validations. These are not validations that you have to be a physiologist by training to learn how to do. You can learn these in a week, I would say. Of course, you need good lab skills and you need to be careful, but they're really learnable processes. So I really encourage everyone to also find a good training in analytical validations if you're interested in measuring hormones in any matrix. Okay, and
we have two more questions. So one from Katarina, and she wants to ask a question about hair cortisol. And her question is it necessary to store the samples in a fridge, and does hair color can affect cortisol concentration?
So I have to say I've never measured cortisol in hair, but I have colleagues here who are experts in hair cortisol. It's not necessary to store the samples in a fridge, but it's necessary to have them in some stabilized form. So there's some processes after you've collected the sample to I would say have it in an appropriate form before you process it. And then you have to do some grinding and some processing of the sample. But it helps, but it's not necessary. There are people who also collect hair cortisol in field settings, and they don't always have access to a fridge right away. And I think, remember that the steroid hormones are really more stable. And so in general, with steroid hormones like cortisol, in any matrix, you don't have to be so concerned about that. It's best practice to have them in a controlled temperature, but they will be okay, at least for a few days, without being stored in fridge. I do not think that hair color affects cortisol concentrations. I can't imagine how it could, but I think there is actually a study from my colleagues showing that the darker hair at least had some different properties. I don't remember, but that would be something to look into. I'm sure there's publications on that. I don't remember myself. Maybe Helena knows.
Yeah, I don't know much more, but I do remember seeing a study where they did find out that that in black and white hair, the cortisol does accumulate at a different rate. And because it has something to do with how much melanin there is to produce the color, so they interact somehow, the molecules.
Thank you
And
both.
then we're at the ending time, but I'm happy
Yeah.
to stay and take more questions if people
We
want
have
to stay.
one-
Whoever wants to leave, you're free to leave. And thank you for being here. But I can also, of course, answer more questions. Neutrophil to lymphocyte ratio for chronic stress. So these are more, I would say, immunological measures. They relate, of course, to homeostasis and in general, to the glucocorticoid stress response, which can have a lot of effects on the immune system. But as an indicator for chronic stress, it can be a great one, but I wouldn't do any indicator in isolation. I would have this measure along with cortisol, along with behavior. So unfortunately, I don't think there's one great indicator that does it all. I think the physiology is so complex, and we need to bring in definitely immune measures. That would have been the third speaker here, to have an immunologist here talking about those indicators, and other physiological measures and behavior. Okay, I don't see more questions coming in. So thanks a lot, everyone, for your interest, for being here. It was a lot of fun. And thank you again, Gina, for organizing. I don't know if you want to say something at the end or.
No, thank you for thank you for this very interesting workshop webinar. I'll just say that we recorded everything, and we will provide every material by mail to every participant, but also it will be on the YouTube channel. If you missed something or if you had to leave earlier, everything will be available online.
And we'll get the PDFs to you today, so you can also give the PDF.
Perfect, yeah.
Helena, could you just stay on for a minute? Or, oh no, never mind. Or is it okay, Gina and Helena, to stay on?
Yes,
Okay.
no worries.
So thank you, everyone.
Thank you, everyone.